yesterday, and this is Dan Davidson, and I work with the Illinois Soybean Association, and I want to thank you for coming online to listen to our webinar on Boron Nutrition for soybeans. I just want to cover a few facts, and then we'll get into the webinar shortly. I want to thank Dr. Nathan Slaton for being our presenter today. Dr. Slaton is Professor of Soil Fertility at the University of Arkansas. You know, some of the duties he pays attention to, he manages a number of the labs that the university has. Also been very involved in fertility on soybeans, and has spent a part of his career working on this boron problem that existed in Arkansas, and they were able to uncover it and develop some recommendations. And Dr. Slaton will talk a bit about that this morning. Again, I want to thank you for coming online. This webinar is being sponsored by U.S. Borax, who markets boron into the ag retail business, and also on behalf of the Illinois Soybean Association and the Soy Advisor, which is a soybean management website. I want to thank them for sponsorship of this event. Just a few things on the logistics. As a CCA, when you register for this event, you would have put in your CCA number, and after this event is over, the CCAs that attended, that number will be submitted to the CCA program for credit. If you listen to this recorded event, that will be posted on the Soy Advisor website, and you want to gain the one CEU in crop management, just log into the CCA site under your name and password, and you can self-apply for credit for this event. A few things on logistics. So with this quick introduction, and during the course of the event, you can submit questions through the panel to your right. And when Dr. Slaton gets done with his presentation, we will take some time to go through these questions and then wrap up promptly at the hour at 11 a.m. this morning. So again, on behalf of the Illinois Soybean Association and U.S. Borax, thank you for being online today. And Dr. Slaton, I'll turn the presentation over to you.
All right, thank you, Dan. And I would like to also welcome everybody and certainly extend my thanks to Dan, the Illinois Soybean Association, and U.S. Borax for asking me to give this seminar to you today. So what we're going to do today is we're going to discuss, kind of what we've learned here in Arkansas, and hopefully you'll be able to take some things back for wherever you're located in soybean production, and how boron, either how to assess whether you've got a problem, and certainly maybe how to recognize boron deficiency if it is occurring. All right, so the outline today, we're just going to start off reviewing a few fundamentals of boron availability and nutrition of plants in regards to plant uptake and soil. We'll spend a little bit of time looking at some of the pictures that we have taken over the years, the past 15 years approximately, of boron deficiency and a little bit of toxicity of soybean here in Arkansas. We'll talk about diagnosing boron nutrition of soybean with tissue analysis. We'll talk about the methods and sources of boron fertilization that are available today. And we'll look at some Arkansas-specific research looking at tissue, potassium, and boron ratio, interactions, and yield response to different boron fertilization strategies. So just to get started, in the soil, boron exists as kind of a neutral element, is HbO3, and this is the format that it is mostly taken up by plants. The boron can be leached from the soil, or at high pH it can be absorbed to certain components in the soil. One of the things about boron is that as soil pH increases, its availability decreases. So it is most likely to be deficient on high pH soils. And again, one of the things that we have seen sporadically, because there's not much lime that is applied here in Arkansas, but occasionally we do see boron deficiency flared after a lime application. We also know that, historically, when you look across the world, and certainly in areas where boron deficiency is a problem, that it tends to occur more frequently on low cation exchange capacity soils. And these low CEC soils, or sandier type soils, also tend to be low in organic matter. And organic matter is a good reservoir or supplier of boron for plants. In regards to the plant, the functions that boron serves in the plant is that most of the boron is actually found in the cell walls. So it's very important in cell wall formation as well as xylem for tissue formation in the plant. It's important in nitrogen metabolism and stomate opening and closing, which is in relation to potassium transport into the guard cells. And a couple probably of the things that I'll key on here and to pay attention to when we start looking at the deficiency symptoms, just as we said that a lot of the boron is included in the cell walls, it is extremely important in cell elongation. And this is one of the things that we see, oddly shaped leaves or growth distortions that appear to be, almost like sometimes slight 2,4-D injury symptomology, or stems that are curved, like say oddly shaped leaves. And this all comes into play in its cell elongation. The other thing that a lot of people have keyed upon with research as well as production type strategies is that boron is key in pollen viability and pollen tube elongation, both of which are essential for proper fertilization and production of seed in crops. So a lot of people have tried to time boron fertilization as a yield enhancing strategy to retain flowers or to increase yield potential by applying boron shortly before flowering or during flowering. One of the other things to key on here is whether boron is mobile in the plant. And you remember back to your soil fertility or CCA training, you know that immobile nutrients, their deficiency symptoms show up in the younger components of the plant or the growing point, whereas mobile elements tend to show up more prominently in the older leaves. And this is going to be true for soybean, that we tend to see the most pronounced symptoms in the youngest growth. One of the other things about boron, and this kind of varies, if you look at 5 or 6 different lists of boron susceptibility of different plants, you may see slightly different versions from one source to another, but certain crops are more susceptible to boron than others. And as a generalization, you tend to have the grasses, and if you look over here into the tolerant category, you see barley, oats, rice, rye, wheat. All of those are grass-type crops, tend to be less susceptible to boron deficiency and have lower overall boron requirements. If you look at their tissue concentrations, typically grasses have lower boron concentrations in their tissue than do dicot plants like soybean or cotton. Over here, the other thing I would point out, in the plants that are considered to be most susceptible to boron deficiency, you can see that there's a few legumes. Alfalfa, clover is not in here listed, but it certainly in the middle category. Peanuts, more susceptible than other plants. Soybean and beans, on the other hand, also legumes, but they are considered to be relatively tolerant. Our experience in Arkansas is that, at least with soybeans, this would not be the case. Soybeans and rice are grown in rotation here quite commonly. We do not see boron deficiency in rice, but on those same fields, we can see complete disasters occur if boron is not applied to the soybean crop. So just a little bit of history here for Arkansas. I started this current position that I'm in 2001, and that particular summer, I've been in Arkansas for nearly 30 years at this point, but In the summer of 2001, we started getting lots of calls from consultants, county agents, and growers. You know, something was occurring in many of the soybean fields, odd growth. You know, many thought it was perhaps a virus or even herbicide injury. Most of our soybeans are grown in the eastern third or the eastern half of Arkansas, with the western part being kind of mountainous, and that's where we grow our poultry and hay crops and pasture. We also have some soybean production in the Arkansas River Valley, which kind of cuts diagonally across the middle of the state. These different colored counties, we kind of did a survey. We didn't hit all the counties, but the ones that we did kind of corresponded with where we were seeing the greatest problem and getting the most calls from our growers, and that was in northeast Arkansas. The colored counties here, Jackson, Woodruff, Craighead, Poinsett, Cross County, are all up in this region, and most of the problems that we were seeing were in the area south of Jonesboro and west of Crowley's Ridge, which kind of splits these counties and runs down kind of parallel to the edge of the state. So this is a hot spot, not just for boron, but this tends to be a hot spot for many of the nutrient deficiencies that we see in Arkansas. This is kind of our canary in the coal mine here in Arkansas. When something's going to go wrong nutritionally, it's going to go wrong here first. The other thing that I would just mention is that in the 60s and early 70s, of course, this is the Delta, so there was a lot of cotton grown in these areas, and there's still, you know, is cotton produced here today. But these are some of the first areas where boron deficiency of cotton was recognized, and has been, you know, boron fertilization of cotton has been a standard practice now, you know, for many decades. But that was kind of across Crowley's Ridge and not so much in this area, which really did not have a history of boron fertilization. So now we're just going to kind of walk through. This is probably the interesting part of the talk, actually, because there's lots of pictures. What does boron deficiency look like? And I'm going to kind of show you the different stages that we have seen boron deficiency here in Arkansas. The picture here on the left, this is soybean that was no-tilled into rice stubble. This was also, you can see that the soil at the time we took this picture is relatively dry. And that's going to be one of the common things that I'll tell you. Boron deficiency here in Arkansas is more common in dry years than in wet years. And these are irrigated soybeans. You know, most of our soybeans are either flood irrigated or furrow irrigated, with also some center pivots, but not so much not many center pivots in this sensitive area that we looked at on the previous slide. These are soybeans that are at probably V2, V3, V4 stage. Really just came out of the ground. They came out of the ground boron deficient. You see that the leaves are kind of oddly shaped. This is not a characteristic of this particular cultivar. This is boron deficiency that you're seeing here from the bird's eye view. And the nodes, what few nodes there are on the plant, are stacked up. And interestingly enough, this particular field, this was after we had started making boron recommendations. And you can see some whitish material here on the soil surface. This particular grower had blended a granular boron product in with his pre-plant P and K, and, you know, broadcast it onto the surface. And as I say, this, you know, it was relatively dry this particular year, and there had not been enough moisture to dissolve the boron fertilizer that was placed on the soil surface. So, and we saw this in several instances, and this is a problem in dry years when the fertilizer is broadcast to the soil surface and there's just not enough dew or rainfall to dissolve it. Here on the right, this is actually one of the tests. You know, if I put a test in 100 producer fields and 99 of them, I'll be about 10 foot away from the problem. But in this particular field, we got our test exactly where it needed to be. You see the beans here on the left-hand side. This is beans that received boron either pre-plant or at an early vegetative growth stage. Lighter green growth, you can see that they're growing and they appear a little bit taller. Beans on the right had not yet received boron, and you can see they've kind of a little bit of light green growth in here, but for the most part they have a darker color. And this is the onset of boron deficiency. The beans had probably been irrigated maybe once at this time, and again, you can see that these beans over here are starting to take off and really grow. These beans, not so much. And like all good folks, you know, we've got a field full of county agents, growers, and consultants out here taking a look at the problem and trying to learn along with us. This was in the early stages of our research. Up here, you can see some of the research data from this particular trial. That boron that we applied foliar from 0 to 2 pounds of boron, we were using Solubor at the time, the effect that the fertilization treatment or boron deficiency had on the number of nodes and the effect on plant height. I would not recommend applying 1 to 2 pounds of boron foliar because you do light the plants up and get quite a bit of foliar burn from those rates. This is just another picture of the color difference between the boron deficient plants, which are the dark plants, and plants that are probably also boron deficient, but are they've got just enough boron that they continue to grow. Once you've seen this and been out in the middle of it, you can spot these fields driving 60 miles an hour down the road because that dark green appearance it really just jumps out at you. One of the other things that is very prominent, and again, most of these soybean fields in this area are rotated in a 1-in-1 rotation with flood irrigated rice. And most of the soybeans, at least in the past, we're moving more to furrow irrigation today. But in the past, a lot of the soybeans, levees were pulled in the beans and they were flood irrigated as well. This is a soybean field that you can see the previous year's rice levee in. Between the levees, and you can see here is a levee, and here's the close-up, the distance between the levee, which would have been the paddy area, very uniform boron deficiency really across this whole field. But where the previous year's levee was formed, especially where the disc of the levee disc go deepest, the soybeans are actually growing. And again, these soybeans would have slightly higher tissue boron concentration, which is why they continue to grow. But they too would probably be deemed low in boron and deficient according to the textbook values. So this is just one of the things that, again, kind of throws up a red flag, the pattern, which is a tillage pattern and rooting depth. Here in Arkansas, most of these silt loam soils have 3 or 4 inches of topsoil, and that's about it until we hit a hard pan. So we're very shallow rooted. We have limited water holding capacity in these soils, and all of these things contribute to the boron problem. This is just another picture of probably one of the southernmost boron deficient fields that I've looked at. And this particular field was irrigated with reservoir water, meaning that between well water irrigated fields from the alluvial aquifer and reservoir or surface water sources don't have calcium and magnesium carbonates dissolved in the water, whereas the water from the alluvial aquifer does. So when we irrigate, as much as we irrigate here in Arkansas, well water tends to increase our soil pH. So the norm for fields that are rotated with rice is that pH is between 7 and 8.2. This particular field, which was irrigated from a local lake or a reservoir, had an acid pH, and the farmer had limed it, and the lime application ended up triggering the boron deficiency. But you can see here, again, this is the previous year's rice levee, where we have the deeper cuts from the levee disc and in between where the levee was formed and not so deep of tillage, you see the severe stunting. This is a close-up picture of a boron deficient plant. And you look at the field in the background, the field itself at this stage when this picture was taken, The healthier looking plants were over waist high. This is an early group 5 maturity group soybean. This plant here, would probably be less than 12 inches. And some of the close-up symptoms, you can see swollen nodes, stacked nodes. The terminal growing point has died, and the plant is trying to regrow with what little boron it can get whenever probably it's irrigated. So it's trying to put some shoots on from these lower nodes. Very few pods, and the pods that typically do form have 1, 2 maximum seeds in it. And you can see the oblong-shaped leaves. And the other symptom you can see up here, it really looks like some herbicide symptomology here with the purple venation on the underside of the leaf. But this is truly boron deficiency. Some closer up looks at the leaves. This is a plant that has been under, I guess, slightly less boron deficient than the one you saw before. But the leaves end up taking on a leathery kind of appearance. You will see some darkening, as well as some chlorosis around the edges of the leaf. And you can also see as the sunshine is coming through this leaf, it's a little more transparent. The leaves, veins, kind of reminiscent of manganese, but again, this is not manganese. The key diagnostic here for boron, one of them anyway, is that the terminal growth will die. And there's really 2 nutrients that will do that: calcium deficiency and boron deficiency. And trust me, these soils are loaded with calcium. Their cation exchange capacity ranges from, say, 9 to 16, and there may be as much as 4,000 or 5,000 parts per million calcium in some of these soils. They're mildly calcareous, but we get lots of calcium and magnesium deposited from the alluvial aquifer. So when you see this terminal growth dead, that's a clue. It's either calcium or boron, and when you know the soil is loaded with calcium, that eliminates it and leaves boron as the cause. And eventually that dead terminal will just completely fall off the plant. It's still hanging on there, but if you touch it, it'll fall over. And if you're in the field looking at these plants and don't know what you're looking at, it almost looks like an insect or something has come through these plants and completely just nipped out or cut off the terminal bud, which you'll find laying on the soil. But that is, again, callus tissue that's forming there, and the terminal growth is just falling off. This is just a field-wide picture. You can see, again, growth variation of a furrow-irrigated field, kind of small growth here, better soybeans here. And one of the other things that we tell our growers to look for, this is, again, one of our trials, and there was very minimal response yield-wise in this particular field to boron, but the plot that you're looking at right here center in the picture is a no boron plot, and the soybeans around it, with the exception, here's another no boron plot, here's another one, and then you can see the field surrounding our test. The plants that did not receive boron retained their leaves longer than where we had applied boron. So, I'm not saying this is going to happen in all fields, because obviously this is a boron nutrition related response, and clearly our tissue analysis did show that the plants were low in boron, despite the fact that we didn't get huge yield response here. There certainly is a maturity response, and this is one of the things that when growers are riding the combine, that they can look out for. Field areas that are retaining their leaves. Obviously, there may be some other factors besides just boron deficiency that can cause that, but certainly boron deficiency or low nutrition is one particular cause, and we'll talk about, if you do see this, how you might be able to diagnose that as boron deficiency at this stage of the game. So whenever we started seeing these problems in 2001, we did a lot of diagnosis, meaning we did field visits, we took tissue samples, we took soil samples. This is just an example here of beans. We always collected comparative samples. BS or B-SUF stands for plants that looked healthy, boron sufficient, and B-DEF stands for plants that appeared to be boron deficient. And you can see that 20 parts per million is what most textbooks call the sufficiency or critical concentration in soybean. And the plants that looked healthy were right at that level. The plants that were showing boron deficiency were at 8 parts per million. The other thing, that's kind of tricky here, if you look at this, the boron deficient plants had lower concentrations of nitrogen, phosphorus, and potassium. Calcium and some of the other micronutrients were higher, but certainly, this might, ask you or cause you to ask the question, well, what is the most deficient nutrient here? And clearly, again, it's boron, because it's furthest below the critical concentration, and the symptomology matches boron. So really, tissue is the way that we would suggest that you diagnose or even evaluate whether boron might be deficient in your fields. Soil test, we were kind of optimistic whenever we started troubleshooting these, that in most of the fields we did find higher Malic-3 is the test that we use. The Malic-3 extractable boron tended to be higher where the soybeans looked better. The problem is both of these numbers are pretty dadgone low, and we have never really been able to establish soil test boron as being a reliable indicator of fields where boron would result in a positive yield response to soybean. So basically, how do we identify boron deficient soils, in Arkansas? Well, we tell the farmers, you need to look at the history of the geography of where you're at. Again, if you're on the west side of Crowley's Ridge in Craighead, Poinsett, Woodruff, Cross, Jackson counties, there's a good chance that, your soybean might respond positively to boron fertilization. You also have to look at, obviously, your cation exchange capacity and your soil pH. So we ask farmers to look at all of these things in determining whether or not they need boron. And if they're in doubt, again, spend the money on a $15 or $20 tissue sample during the season to identify whether, your fields appear to be sufficient or deficient in boron. But right now, we do not rely, and I don't recommend anybody rely on anybody's soil test for boron to indicate whether or not the crop would respond. I know there are some other soil tests out there. The hot water extractable boron is used in many labs, but there's a lot of deviations from one lab to the next. And if they cannot produce a yield curve showing the crop response to boron fertilization across the different amounts of boron that is extracted by the method, I really think that, that's kind of risky business of spending money on fertilizer that may not be warranted. I do think that there is potential to identify soils that have excessive boron and might lead to boron toxicity, provided this is not a fertilization mistake. But clearly, I think that we can identify soils that are excessive in boron. So what about boron toxicity? We've been recommending boron now in Arkansas for almost 15 years, and I've only looked at one field that has what I've decided was boron toxicity. And the history of this field, which you can see the pictures of the symptoms here on the left-hand side, these fields were huge, probably a half-mile long, a couple of 100 acres in this field. And you can see the necrosis along the edges of the leaf tended to be worse kind of down in the canopy than on top. And you can see this picture was taken in 2012. We tried to duplicate these symptoms that next winter in the greenhouse, growing some soybean in pots. We put boron out based on the surface area of the pot and expressed it here in pounds of boron per acre. And you can see that, and this was soil not from this field but from this general area, that even a 10-pound rate of boron, which would be probably a 10x rate of what would be recommended pre-plant, we really got minimal increase in tissue boron of the soybean. But when we jumped it up to 40-pound, we did start seeing the same type of symptoms, and the tissue or leaf concentration of boron in the plants really jumped up at that stage. So we never did quite figure out I really thought that this was a dealer error where they had just simply blended too much granular boron in with the pre-plant P and K. But when the farmer looked at what he paid for, it was supposedly 1 pound of granular boron. Now whether or not there was a mixing error that nobody ever owned up to, we don't know. But one of the interesting things, again, what we see in Arkansas is that this is well water irrigated field, and because they've been watered from the same entry, the water is entered at the same entry point for decades. We see pH gradients at the top of the field. The pH tends to be highest, and as you move downhill to the lower elevations of the field, there's a pH gradient with pH decreasing. The symptoms of this field were worse in the lower end of the field where the pH was just in the upper 5s. There was no symptomology on the upper end of the field where the pH was in the upper 7s. So again, clearly this was associated with the lower pH areas of the field, and I just find it difficult to believe, based on what we have seen, and I'll show you some data in a minute, that these type of symptoms would have been caused by a normal fertilizer rate. So how much boron is too much? Well, this is when we first started this. We'd read the textbooks and seen the statement that there is a very narrow window between boron deficiency and boron toxicity. So that was a concern to us before we started recommending boron widespread across eastern Arkansas. The literature would suggest that for soybean, 20 to 60 parts per million is the sufficiency range. And so if that's the sufficiency range, anything above 60 would be considered high. So at some point above 60, you have a toxic concentration. And I don't know that we have that well defined. We certainly have not tried to define that here with our research, but I honestly believe it's well above 100 parts per million. The picture that you saw on the previous slide, was in the 200 to 350 part per million range before we started seeing those type of symptoms from the field. In this research, you see this is soybean yield on the left-hand side of this figure. The different color bars represent different pre-plant applied granular boron rates. Pretty good yields starting off, and we did see a tendency for yields to start to decline a little bit as the boron rate increased to 6 and 8 pounds per acre. But there were no symptoms of boron toxicity in this particular trial or a second site with a lower pH where we did this work. However, we did see a relatively good increase in leaf boron concentration with a fairly substantial jump between the 6 and 8 pound rates. Kind of a plateau between 1 and 6, not a huge amount of increase. So again, I think gross over fertilization errors with boron would be a concern, but a little bit of extra boron I don't expect is going to cause a huge problem, especially on soils that already have neutral to alkaline pH. So just a review before we move on, where we would expect to see boron deficiency is low organic matter soils. Most of the soils where this problem occurs have organic matter between 2 and 2.5% in the top 4 inches. Relatively low cation exchange. Certainly sandy loam soils that have CECs below 10 are very prone to boron deficiency, but even the silt loam soils, which for Arkansas, soils that have 20% clay and where rice and soybean are grown, routinely have cation exchanges in the mid to upper teens. pH greater than 6.5. Recent lime applications when applied to acid soils. Shallow soil or rooting depth are also triggers. And the big one for us in Arkansas is really dry soil conditions. When we have above normal rainfall, reducing the need for irrigation, we see very few true boron deficiencies. When we have hot, dry years that start, right after soybean is planted, that's when we see the greatest number of boron problems. Since we've been applying boron for 12 or 14 years now, the number of deficiencies have also decreased, which suggests that there's probably a residual component to fertilization as well. So one of the things that Daniel, when we were talking, asked me about was, this boron and potassium interaction. And I really hadn't ever looked at much of our data in regards to this. I'm certainly aware that the literature, talks a little bit about aggressive potassium fertilization, possibly increasing a response to boron. But I'd never really looked at what we had seen in the plant. So we went into one of our short-term fertilization trials, and by this I simply mean it was a field where we put boron out, and it was a one-year trial and we were done. On the left, again, these bars represent different potash application rates in pounds of K2O, and you can see that as our K rate increases from 0 to 160 units, leaf boron concentration decreased about 3.5 parts per million. On the other hand, leaf K concentration increased quite a bit. So this is a short-term trial. What about a longer-term trial? And this is, again, a similar silt-loam soil, but one that actually tends to fix K. And this is the 15th year of this trial where these rates have been applied annually. So again, we see pretty strong boron concentrations in the tissue, and that's because we apply boron at this particular site because it's in that geographic area where it's a problem. But again, we see that as potassium fertilizer rate increases with uniform boron fertilization, tissue boron does decrease. And when you look at leaf concentration, we're seeing a nice response here to our annual potassium fertilizer rates. Yield-wise, this site is extremely responsive to potassium fertilizer. This is, again, the 2015 yields. And when you look at the leaf potassium to boron ratio, again, you can see that as our K rate increases, that ratio becomes wider. I guess the thing I would say about this is that I still think that the leaf boron concentration is the first thing to look at in this regard. And as long as your leaf boron concentrations are deemed adequate, then I have no problem, because whether it was a short-term trial or a long-term trial, we did not see a decrease in leaf boron concentration in all of the trials that we have conducted. But where we did see it, the decrease in leaf boron concentration tended to be less than 3 to 5 parts per million. So let's just review a little bit about how the plant takes boron up. This is information out of the Illinois research program. Fred Below's program, I believe. This is Ross Bender, who's now with the Mosaic Company, and some of his dissertation research at the University of Illinois, showing boron uptake curves and distribution among different plant parts. The key things here I'd want you to look at is the fact that, this is a late 2, early 3 maturity group. And if we just look at the R2 growth stage, about 25% of the boron had been taken up by these early maturity group plants at this stage. Compare that to the total amount of boron uptake during the year, which was about 4.5 ounces, or a quarter to a third of a pound of boron in the above ground portion of the soybean plant. Now, we've done some similar work here in Arkansas. You have to forgive me, I did not convert these figures from metric to English units. But again, the total uptake, if you were to convert that, would be about 0.2 to a third of a pound of boron per acre. The other thing I would alert you to is these, we're growing late late 4s and early 5s to mid 5s here in Arkansas, which, you have more growth before they hit the R2 stage. So in Arkansas, we're seeing 35 to 40% of the boron uptake before we hit flowering and pod set. And again, this is just simply the difference, I think, in maturity group. But clearly, for both of them, it shows that the majority, at least 60 to 70, 75% of the boron is taken up during reproductive growth, which I think it's been said that, there's some elements, copper, boron, are examples where the plants might be able to grow just fine vegetatively, but reproduction and seed set would be problematic due to the functions that these elements have in seed development and pollination. So some of the Arkansas experiences, I guess we could summarize for tissue analysis. We tried to use tissue analysis early season, V3, V4 stage, to identify fields that might respond to boron. And basically what we showed is that depending upon the moisture content, we saw a lot of variability from sample times all the way up to the R2 stage. So we never felt really good about being able to predict whether or not we were going to get a response from an early vegetative tissue sample. What we have found is that, in all of the research where we have routinely collected samples, again, from late 4, early 5 maturity group soybeans, is that at the R1, R2 stage, the tissue boron concentration has been a fairly good indicator of whether or not we would see a yield response, whether it was marginally significant or truly statistically significant. I do feel pretty good about the textbook value of 20 parts per million as a critical concentration, and I'll show you a little bit of data on that here in just a minute. But our experience in troubleshooting these problem fields is that you're probably not going to see true deficiency symptoms until your concentrations are less than 12 parts per million. And by this time, you're probably already losing some yield potential. And once the deficiency symptoms occur, you're definitely losing yield potential. The one thing I would stress, not so much for boron, because again, I feel pretty good about this number, but when you start deviating from the R1, R2 growth stage for boron, and perhaps even the critical concentrations that are listed for some of the other micronutrients, most of those critical concentrations are based upon a survey rather than hard research data where they looked at tissue concentrations and compared that to a yield response. And that's just because a lot of the micronutrients, there's not a sufficient amount of deficiency out there to be able to quantify that relationship. So in Arkansas, all the trials where we have collected leaf tissue at the R1, R2 growth stage, this is kind of what the relationship looks like. This is the relative yield of the untreated check, meaning plants that received no boron. At this particular trial where I'm pointing, produced only 45% of the yield of the maximum yield produced by a plant or a plot that received boron. So what we see is that the area I have shaded here, 20 to 30 parts per million, this is probably a pretty good estimate of sufficient boron for nutrition for soybean plants. Anything below that, and we're probably starting to lose a little bit of yield potential. And that's why, again, I'm encouraging you, before you just go out and start throwing boron around on your fields, assess, the potential need for it. And I really feel like, if you think at R2 stage, that you're in this 15 to 20 part per million range, you can get a good yield response to boron at that time because the plant hasn't bottomed out. But if you're up here at 30, 35, 40 parts per million, there's no need for boron. We have found that the seed can be a pretty good post-harvest indicator. When your seed is less than 5 parts per million boron, you were definitely deficient and you would have seen a yield response. And this is kind of what we see with boron deficiency. We see shriveled seed, a lot of seed abortion. So a lot of this seed, as a matter of fact, is probably going to pass through the combine and never going to make it into the bin. This is compared to boron sufficient or fertilized soybean on the right. If you're 5 to 10 parts per million, yeah, you need to take a look at your program, and maybe some type of boron fertilization is warranted. But give it some thought. Talk to somebody who has some experience here before you jump into it whole hog. Seed that has greater than 10 parts per million is probably normal. We took some of the seed from many of the trials that we conducted, and we looked at seed quality, because in this area where in northeast Arkansas where we see problems, we do tend to have some seed production, and you can see that the seed boron concentration can be fairly low, and as we increase boron application rate, our seed concentration increased, and the seed concentration increase was greater when we put it on later in the growing season than early. When we took this seed from one of our most responsive sites, we did see that germination and accelerated aging were very poor. So this would not be good seed stock. This is just kind of an assessment of our early production or our early trials. The black bars indicate statistically significant yield responses to boron fertilization. The blue bars were certainly showing a trend of marginal response. The white bars were lower pH less than 7 areas that we did not expect to see boron positive response to. This site here that was most responsive did not show any symptoms until pod set. And these were July planted beans that the untreated control kept 15 bushel, and where we had applied boron by the R2 stage, we were cutting in the mid-30s. So a great response here. What are the options? Well, pre-plant we have granular boron sources, and there's several of them out there. I don't know that they're all created equal, but they are out there. They can be blended with your pre-plant fertilizer, P and K, and distributed across the field. And then you have, potential segregation as well as distribution problems from particle densities. And typically we would apply 1 pound of boron per acre in this manner. One of the more recent developments, the Mosaic company has a product called Aspire. It's 58% K2O and 0.5% boron. I'll show you a slide of yield data on this in a minute, but it has been a fantastic product for us here in Arkansas, and I think it is something to look at. The other thing is you can put boron solution out with, say, a pre-plant herbicide or a burn-down herbicide, but one of the problems we have here in tank mixing with herbicide, whether it's going out pre-plant as a burn-down or post-emergence as a contact herbicide, you got to be careful that you're not antagonizing activity and reducing weed control, especially for weeds that, like glyphosate, may be marginal on to begin with. Manure can also be a source. We haven't done any boron fertilization work with manure, but about a ton of poultry litter contains about 1/10 of a pound. If you're going to do it post-emergence, timing can be critical, because, again, we can have very early season boron deficiency. And typically, what I tell growers is go on the lower end of this scale. 1/10 of a pound of boron can go a long way because, again, the plant, fairly high-yielding soybean is only taking up about 1/4 of a pound of boron per acre, and if you got good leaf area, you're going to intercept a lot of that spray to begin with. This is the summary of 4 different trials at one of our stations where boron deficiency can be a problem. We see good responses to potassium fertilization here, and compared to 1/2 to 1 pound of boron applied pre-plant, depending on the study. That's why I've got a range in here. The Aspire product showed an excellent response. There is a residual response of boron in these fields. This particular slide shows the soybean tissue boron concentration from the first year of boron application. This would be no boron, no additional boron was applied, and rice was grown the second year. These values are multiplied times 10 just to be able to see the differences. And then again, you can still see that the third year after application, soybean that were grown in year 3 still have elevated boron concentrations where boron was applied. And lastly, we stressed our growers in the areas where boron is a problem, prevention, because this is the field where the levee, the previous year's rice levee showed up very prominently. We had 2 untreated checks because we knew we were going to have a lot of variability, and we applied boron on a weekly basis, and we had 6 reps here, and basically there was no response. Once you have boron deficient plants, there is no guarantee that supplying boron will aid in their recovery. We've never quite figured out, what is the drop dead recovery and when soybeans will recover, but certainly it's a gamble. So that's why we stress prevention rather than rescue type applications. To summarize, we really feel tissue analysis can be used to evaluate boron deficiency or nutrition in your fields. We like doing this at the R2 stage. For further north, you might want to wait until a full R2 stage to allow those beans to get just a little better root system than if you're just taking assessment type tissue samples. I understand that the textbook critical concentrations for different micronutrients may or may not be valid. We feel like the one for boron at 20 parts per million is pretty good. And if you're wondering about post-harvest, reasons for maybe low yields and odd growth, and you saw those leaves hanging onto the plant, seed analysis can also be used. I'm not necessarily an advocate of blanket fertilization with boron just to try to produce ultra high yields. Certainly our data shows that a little boron goes a long way. And there are a number of different fertilization methods that can be used. You just need to find the one that best fits your system. And I would like to recognize again that the research here has been funded by Arkansas Soybean Checkoff, Rice Checkoff funds, fertilizer tonnage fees. U.S. Borax funded some of the early work with, especially with rice here in Arkansas, which we found not to respond to boron. And lastly, I'd like to thank Daniel and the Illinois Soybean Association for inviting me here. With that, hopefully we've got time for a question.
Nathan, wow, as an agronomist speaking, quite a webinar. A lot of information you covered. Very, very thorough. So thank you very much. A couple questions here. We got 5 minutes for some questions here. So we'll talk a bit about boron mobility in the plant and how that relates to managing boron.
Well, as you saw in that one slide, just from the different time applications of boron, early vegetative growth at the B3, B4 stage compared to boron applied to a bigger plant at R2. There was not a whole lot of translocation of boron from the leaves from the B4 application to the seed. So boron is immobile in the plant. That's why, collecting tissue samples from one of the top 3 nodes of the plant I think is critical, because you can get some gradients there in the plant. So at least for soybean, boron is considered to be immobile. And that's why, if you're concerned about seed boron concentration, trying to increase it, a later season application might be beneficial. But what I did not show is that we took that seed and replanted it, and there was really no benefit from elevated boron seed concentrations to the plants that originated from that seed. So long as it would germinate, it produced good plants.
Okay. So you talked about this application, you had that one slide about the toxicity. So if somebody's kind of doing a blanket application like 0.5 to 1 pounds in a granular broadcast with other fertilizers, would there ever be any risk of creating that toxicity situation?
Well, my mom told me to never say never and or always, because those are pretty inclusive. I think so long as you're only putting out 1 pound of boron, I don't think that there is a great risk of boron toxicity. However, you still have to look at several other factors. pH of the soil. Low pH soils are going to be at greater risk. Soils that are very sandy are obviously prone to boron deficiency, but they are also, you don't have any of the mechanisms there to absorb or buffer that boron. So when you put boron on a sandy soil, you're probably going to get greater uptake, provided that it stays within the root zone. So I don't think there's a great danger there, but again, I think certainly the textbooks all caution that, boron fertilization needs to be done with some reason and judicious use of the rates. So,
you think about, if you have a 60 or 70 bushel soybean crop, how much actual boron is actually taken out of the soil? We think of NP and K, and 60 bushel soybean crop might need 450 pounds of N. How a 60 bushel soybean crop would take out how many pounds of boron from the soil? Did you ever calculate that?
Yeah, that was shown in these slides here from both Illinois, and I'll leave this one from Ross's work up there. 4.5 ounces is just over 1/4 of a pound. And then
it's about 1/4 of a pound. Okay.
And then you can see the crop removal, which is approximately 35, not quite 40% of that amount. So relatively low amounts of removal.
So someplace around 1/3 of a pound taken off with a 60 bushel yield.
Yeah, I cannot remember what Ross's yields were, but again, the work that we did here in Arkansas, that's where our yields were in the mid to upper 50s, and we had similar values.
Yeah, 0.2 to 0.3. Good. Okay. So, done all this work in Arkansas, you had some problem areas. And so your general kind of recommendation to farmers regarding boron fertility in general for Arkansas in areas where there hasn't been a history of any boron deficiency, what is that kind of recommendation, that management recommendation?
Well, I guess, we have a lot of alkaline soils here, not just our sandy loams, silt loams, but also clayey soils. And I guess what I would recommend is that the farmer simply, before he goes out whole hog, and starts applying boron or including boron in his fertilization program, he can take some tissue samples and simply assess, the sufficiency of the plants that are growing in his fields for a relatively low cost. And that, to me, is the wise way to go. If if the farmer was convinced that he needed it, then again there's a lot of options. You can foliar feed, or you can use some of the newer product, Aspire, or again the old practice of granulobor or granular boron blended in with your pre-plant P and K works. I would just make sure that it was applied several weeks in advance of planting to give that boron a chance to distribute the soil.
So here's one that came here. Would a dry boron source like Aspire, the top dressed on at R2 be of value?
Potentially, yes. I personally have not looked at that, but I know that from a potassium standpoint, we can top dress potassium on up even further than R2 and see good yield responses. So at least here in Arkansas, our beans, because they're surface irrigated and we don't have very deep rooting depth, I think we can get the boron uptake. But again, that's me guessing. I don't have research data to back that up.
You had talked about, I think I want to just reinforce this, that the tissue testing sufficiency is 20 parts per million, but down to 12 parts per million, you'll see no visible symptoms. Down to 12 parts per million, but yet, if you're down to 12 or 15, you don't see anything, but that can still have an impact?
Sure, yeah, and that's, I think, what the textbooks would typically call, hidden hunger. The plant is not necessarily telling you with real loud, messages that it's boron deficient, but you are probably, losing a little bit of yield at that level.
Well, we're here right at 11 a.m., Dr. Slaton. Again, I want to thank Dr. Slaton for giving this presentation today. You covered a lot of good information. I personally have to go back and listen to this again to pick everything up that I missed or didn't understand. So first, thank you very much. Again, I want to thank the Soybean Association and U.S. Borax, part of the Rio Tinto Minerals, for being a sponsor of this event. Again, as a CCA, if you registered, you'll get one CEU in crop management, and if you listen to the recording, you can go back online in your CCA account and self-apply for one CEU. Again, thank you very much. Thank you, Dr. Slaton, and thank you, everyone, for participating in this webinar today, and have a very good day.